Welcome to the Microscopists, a bite-sized bio podcast, hosted by Peter O'Toole, sponsored by Zeiss Microscopy.
Today I'm joined by Kelly Rogers from Melbourne, and Kelly shares her unexpected path from aspiring forester to a leading microscopist. She reflects on her first encounter with a confocal in Hong Kong, and talks about balancing research, leadership and team building in one of Australia's top institutes. All in this episode of The Microscopists. Welcome to The Microscopists. I'm Peter O'Toole from the University of York, and today I'm joined by Kelly Rogers from WEHI, which is the Walter and Eliza Hall Institute down at Melbourne in Australia. Kelly, how are you today?
I'm good, thank you, Peter, and thank you for having me.
Oh, it's a pleasure. I was trying to rack back when it was the first time we met, and it was definitely at Pasteur for sure.
It was.
When you were
it was.
for Spencer.
Yeah.
But I can't remember if it was a conference I was speaking at or whether it was a review of Pasteur. I bet you don't remember.
I think. No, I think I do remember actually. I think I met you. I probably met you on several occasions at the Pasteur, but the one time, I think the first time I met you was probably in 2002, and that's when I was a postdoc actually at the Pasteur. It could have been 2002, 2003, and it was when Spencer hosted ALMI. I think he hosted ALMI that year, maybe. And I think you might have come along to that. Yeah. Was it ALMI? And I think, because I remember there were some other interesting speakers as well at that conference, including, I think, Jennifer Limpenkoch-Swartz might have been one of the speakers, and maybe, I can't remember the others, but it was, I remember Kurt Anderson was there as well, and Spencer always had a great array of people coming along to those events, and it was always really interesting to meet lots of people. Jason Swedlow, I think I met him the first time in 2002. So that was a great time. I was a postdoc, by the way. I was not in Spencer's lab at that time. I was undertaking research in another lab, but I was spending most of my time in Spencer's lab and working with him and his team most of the time.
Do you realise you've name dropped 4 previous guests of the Microscopists?
Oh, really?
Yeah.
But if I think back to that time, which was a really exciting time for me, and I was still a sort of junior postdoc at that time, they're the people who I remember very well that Spencer introduced me to. So it was a pretty exciting time.
I think we'll come to Spencer a little later. So, Kelly, what is your current job? What is your current role?
Currently, I'm the head of the Division of Advanced Technology and Biology at the Walter and Eliza Hall Institute. I continue to manage the microscopy facility, which we call the Centre for Dynamic Imaging. The division that I'm head of is made up of many of the advanced technology platforms at the Institute. So it includes imaging, proteomics, genomics, flow cytometry, various others. There's about there's 7 labs in total. They're not all of the technology platforms are in that within my division, but most of them are. So it's great actually. We're a great they're a great team that I work with, and there's some really exciting things going on, and I think perhaps the most interesting thing that I'm seeing, and you're probably seeing as well, is that there tends to be a lot more crossover now between one platform and another, and particularly with methods like spatial omics. So it starts to become difficult to understand where those technologies should be placed. But we're doing our very best to work together and get optimize those sorts of workflows, and so we're working a lot more together now, I would say, than what we did in the past.
That sounds very similar to my own role.
Yes.
And actually, thinking about those correlative techniques, actually, I had my head of genomics only this morning, Sally, coming up to us, sort of saying, "Pete, we need more help. So we're going to offload this day one of the spatial onto the microscopy team. Okay, so
Yes.
you're my team as well. I said that's brilliant. It's exactly how it would be. And actually, if I saw my latest slide deck on budgets, even how we now we all have the individual budgets, so we know what's going on, but we report back as one. So it doesn't matter where the money's going in; it's coming into the facility, and staff are now. So yes, they'll be paying my staff.
Yeah.
My staff. I probably won't bill for my staff time. Sally will just charge for all the staff time. It's on hers, but it's used my staff. Doesn't matter because we
Yeah.
are more joined than we used to be.
Yes.
The complexities.
Yeah,
But
that's right.
interesting, how many staff are you managing at the moment?
I think there are 10 or 11. Well, I have at least one student in my team. I should know this. I think it's 10 or 11 staff right now, maybe 10, including
Is that just
at
in
least
the
one student.
imaging and the lab heads, or is that across the facilities?
Well, actually, that's just my microscopy, the team in microscopy. There are 6 lab heads that I like to say collaborate with, but
Yep.
that report to myself. Yeah, so that means that it's actually more than 10 or 11 then, because it would extend with those additional laboratory heads that I work closely with who manage
Mhm.
the different platforms in my division.
So it's a big job. That's a lot of performance reviews at the end of a year.
Yeah, and we actually don't just do those performance reviews once a year. We actually do them, we kind of stage them. So the objectives or the goals, individual goals are kind of set at the beginning of the year. And then we do a review mid-year. And then we also review it again at the end of the year. And I guess that creates a bit of continuity, enables people to sort of discuss any challenges they might be having, allows them to report in about any accomplishments that they've, any achievements that they've had. And it works pretty well, but it always feels like it comes up it comes up a lot faster than I
Yeah.
expect. And it's true that I end up having to do about 20 of them. But more recently, I've kind of restructured my microscopy lab, and now I have senior members of my lab who I've been working with for some time who manage their own small teams inside of my lab. So I'm sort of, I guess, so, that means that I'm not actually managing directly all of those people that I mentioned. I'm actually managing the senior research scientists or research scientists officers in my lab, and then they're managing a small team. So as an example, I've got Lachlan Whitehead, who's been working together with
Mhm.
me since 2012, and Lockie, he has 3 people working together with him in the image analysis space. And so he takes care of that group, and there's also another excellent, talented senior research officer in that team who contributes to the management of that team. And then there's Niall Gagan, who comes from University of Glasgow, and he's managing the microscopy team. And then I've got Emma Watson, who is a senior member of my team, and she's sort of looking after aspects of high-plex protein and sort of spatial omics and 3D multiplex imaging. So it's sort of like, I guess there's 3 or 4 areas, and each of those areas, just inside of the microscopy team alone, managed by senior scientists in my lab. So it's great. So it's not that challenging. They do the performance reviews now, not me.
Is this, I can't, because on Zoom, I can only see the thumbnails of the pictures, but is this the team or a lot of the team?
It is the team, and I apologize for that because it's really poor resolution, which is really bad for a microscopist. But I couldn't find the original image, so I kind of just copied it from my PowerPoint slide. So the resolution is bad. But yes, that's the team there. That's our Christmas lunch that we had last year. And so in that mix, there are at least, I think, 3 students in there. And then Niall and Lockie are on the right, closest to us. Niall is closest
Is
to
this a
us
better
with the
picture?
glasses. Yeah, this one's a better
There you
picture,
go.
actually. Yeah. Yeah. And so this is not everybody. There's a few people missing, but most of the people in this group here, there are 2 students, and then there's at least one postdoc who's, because I actually didn't mention that I also run a research lab. Um, and so there's one postdoc in this group here who who's completely dedicated to that research, and that's malaria research that we do.
That's wow. How yeah. Okay, I'm going to ask how you balance research with the facility, but actually, I also have research on the side.
Yes.
How do you petition the conflict of interest? How do you prioritize? What is your priority? And another
Yeah.
academic comes, I need to do this today, and you've got your own grant deadline. How do you prioritize?
Yeah, really good question. If I'm honest with you, we've always prioritized the requirements of researchers first. We've always, I mean, obviously there could be occasions. The one that you just mentioned is a really challenging one to,
And extreme and rare.
uh, yeah, that's a very extreme example. But normally, one of the things that's been hard for us is that we have always prioritized what the needs of research teams inside of the institute first. And so when I started out, I was recruited as a laboratory head in 2017. I started the facility in 2009, but then got promoted to laboratory head in 2017. And that's when I needed to start balancing, what we were doing as a research team versus what we were doing as a platform. And at that time, it was completely mixed. So our research was really heavily based on the requirements of, the challenges that researchers inside of the Institute were facing. So what we wanted to be able to do is come up with novel ways or, innovative ways to overcome what some of the existing challenges are. And that's the direction that our research took. And so usually that was in close collaboration with 1 or 2 or more research teams. And so that's how we were able to balance it. The downside of that is that it's always taken us a lot longer to get our research papers published. Even once all the data is acquired and once all the figures have been created, and right now I've got one sitting on my desk that needs to be reviewed, and I'm the one who's holding it up. But yeah, so that's been the challenge, because in fact we always have prioritized people's needs first before the research lab. But that all changed. That all changed in 2021. Because I was awarded a government grant to do the research that we're doing. And that meant that at that time, I was able to recruit a postdoc or 2 and a research assistant who were just going to be working, committing their time to that project. And that made it a lot easier for us then to prioritize that research. But without that grant, I think we would go back to, once that grant is finished, we'll go back to depending, having to prioritize again, really what we do with research teams at the Institute. But that's how we do it.
Okay. So let me take you back. You could not, I can believe even when you started your postdoc at Pasteur, I can't believe for a minute you thought you'd be where you are today. So I'm going to take you back even further in time to when you were, I don't know, 7, 8, 9, 10 years of age. What was the first job that you ever fancied doing? What was your sort of childhood dream job when you were super young?
Oh, that's interesting. I wanted to be a forester. I don't know why. I wanted to be a forester. I wanted to be working in a national park, to be a park ranger or something like that. And I remember that when I did work experience with someone, but that was once I was in high school, so I was a bit older at that point. I remember them asking me about what I wanted to do, and and they said to me, "But Kelly, aren't you scared of spiders and scared of being alone in the forest in the dark?" And I actually started to think about it because up until that point, I never really thought about the spider factor and the darkness.
Practicality.
And so at that point, I switched and I changed direction, and I no longer wanted to be a forester after that. So that if anything good came out of, I don't know if that was a good thing, but if anything good came out of my work experience, that I would have done in year 9 or year 10, it would have been that they like challenged me about my decision to become a forester, and I realized maybe that's not exactly what I want to do.
So when did your interest in science kick in?
I think I was always interested in science, actually. There used to be this program that they showed in Australia called the Curiosity Show. And I would get home from school and I'd turn on the Curiosity Show and they would do all these various experiments and things. And I would always try and replicate them. I'm not sure if I ever admitted that to any of my friends, but I
You
would get
have
out the
no.
back and in my dad's workshop and I would try to replicate them. But yeah, so I think I was always interested in science. Like maybe I get kind of fixated on things that most others would not be interested in. And I think that was really, it became apparent I was very interested in science. When I went to university to study biology or physiology, I did marine biology initially. I was really interested in marine biology, and but I also changed my mind about that because we had a field trip, and we had a field trip where we had to go out on a boat into the bay, into Port Phillip Bay. And it was a very, very cold day, and it was raining and it was windy. And I think I didn't really have very good jacket on, and I didn't enjoy it at all. And then I remember them saying, most people want to become a marine biologist because they think that you're going to be able to swim with the dolphins. And yeah, I had quite a naive perspective on what that entailed. And then they said to us, but most of the time, what we do is go out into the bay, we drop a bucket into the ocean, and we scoop up some samples from the bottom of the ocean, and we take it back to the lab and look at it under the microscope. And so yeah, I think I moved on from that one too. I realized that's not what I want to do. And yeah, so I eventually I figured it out. What I was really most passionate about and most interested in was any kind of research that sort of leads to, improvements in healthcare or improvements, medical research was where I went after that, because I realized that that's really where my passion was, where I was most interested.
So where did you do your PhD?
I did my PhD at Griffith University on the Gold Coast, which probably I actually grew up in Melbourne, but I moved to Brisbane, and I moved to the Gold Coast with some friends after I completed my honours degree in Melbourne. And so I went, while I was there, I got a job, initially a job at SeaWorld, actually, developing photos. So I was developing, the 35mm films. And that kind of stemmed from the fact that when I was at university, I had a casual job at Kmart, and I was working in their photo lab. And my role was to develop the film and then print the photos. And so I got a job at SeaWorld doing the same thing. And the pay was really, really bad, but I loved it. I absolutely loved it. And I would put on my uniform and skip in. Basically, in my mind, I was skipping in there each day because I loved it so much. And it was a very short stint. Soon after that, I started working at the university on the Gold Coast, and that's what led me into the PhD that I then did.
And then from the PhD straight to the postdoc at Pasteur, or was there another step in between?
No, I went straight to the postdoc at Pasteur from my PhD.
That's a
And
big step
yeah, that was
to
in
go from
2002.
Australia to France. Yeah, from Gold Coast to
Yes,
Paris. That's a culturally quite a big shift. Yeah.
that
First,
was an enormous cultural shift for me. Yeah, it was a big shift. Well, I during my PhD, I actually spent about 6 months, I would say, in Hong Kong, working in Hong Kong because I had a boyfriend at that time who had moved from the Gold Coast to Hong Kong to work, and so I was kind of doing the long distance
Mhm.
relationship, and I was regularly going to Hong Kong, and eventually I managed to organize like a research stay in Hong Kong where I was doing part of my PhD. And that's where I first got introduced to confocal, was at that moment. When I first used a confocal microscope and first used a customized microscope setup for doing ratiometric calcium imaging. Yeah, so that was my introduction into microscopy, actually, was that moment, because I went to Hong Kong after this boyfriend of mine at the time, and that's where I got introduced to sort of, I guess, a custom built microscope setup, and I don't recall there ever being a confocal microscope at that time at the university I was doing my PhD at. So it was pretty exciting to see that.
I've got to ask Kelly, what was it? What was the confocal?
Oh goodness, I have no idea. I couldn't. I don't.
God,
Was it
that
a?
time, ratiometric biorad, surely.
I was going to say it was a biorad. I was thinking to myself immediately as you said that it must have been a biorad, but yeah, I don't remember. It would have been in when I went there. I think it was sort of end of the 90s, around sort of 99, 2000. So
NYC. Oh, it could have been a Radiance, I guess. Radiance out then. I think Radiance was just coming out then, wasn't it? I think. Oh, anyway,
maybe,
that
I
doesn't
don't
matter.
know. It was all new to
That
me at
doesn't
that
matter.
time. Yeah.
So you're from Hong Kong and then to Paris. Again,
Well,
that's
I,
another.
I I was only in Hong Kong. I did go back to the Gold Coast after that, and then from the Gold Coast to Paris. So yes, it was. If I'm honest with you, I found that the cultural shock, if you can call it that, was much greater in Paris than it was in Hong Kong, and Hong Kong was easy. I would say it was a great place. I loved it. Paris was a bit more challenging.
But you stayed. How long? How long? How long were you in Paris?
I was in Paris for about 7 years. So almost, yeah, almost, I would say about 7 years. So a long time. I left there in 2009, and that's when I came back to Melbourne, where I was originally from, and I set up the microscopy facility there because they had this fantastic flow cytometry facility. Um, but they didn't have a microscopy facility. They had some high-end microscopes on different levels that were being managed by postdocs, I think, but they didn't really have a centralized facility dedicated to microscopy at that time.
So going back to Paris, I guess you then moved into Spencer's group at some point.
Yeah, I moved into Spencer's group. So I spent almost 5 years in this research lab that I started in. And most of the time we were working on various projects, developing, different types of techniques. We were, you know, trying to develop an approach to be able to look at neural connectivity in the mouse brain. And I then moved into Spencer's lab. It was in 2007. So, but if I'm honest with you, I probably spent most of my time with Spencer's lab up until that moment. I was always hanging around, like probably a bad smell in his lab. I always felt part of his lab, and he always made me feel like I was part of his lab. And we did a lot of work together.
So you threw in some pictures of Spencer. Oh, I can't put Spencer.
Yes.
So
Yes, that's Spencer. Yeah.
I presume a lab dinner, and you've also got, wow, that's a
Yes,
group
this is
and a half picture, isn't it?
yes, it is. Yeah, you probably recognize a few faces there. Yeah, that was Spencer's lab at that particular time. I think that would have been maybe it was in 2007 or 2008. Um, and so I don't know if you know Olivier Renaud. I think he's now at the
Yeah.
Curie Institute. Yeah.
Yep.
Yes, that's right. Yeah. Um,
And you sent
I
it home.
just yeah, so
Wow,
this is
what a
in
picture.
2002. This is very, very early on in 2002, and that was Spencer and Raphael, who both Raphael worked in my research lab, and a couple of other friends who were also expats in Paris. One from Sydney, Bronwyn, who's on the right there, and she was at I can't remember which university she was based at, but she was a biologist as well. And that was that's a funny story actually, because I wanted to include this photo because I remember that we went to the restaurant, um, and I think it was a dinner for my birthday actually, and we all ordered our meals, and I ordered without realizing, I think it was sheep kidneys or something like that, because I thought it was just lamb of some sort. And when the guy asked me, how would you like it done? And I said medium, I think I said medium, and he looked at me with a big surprise and he said, really? Medium? And I said, yes, yep, yep. And I had it completely wrong. So they brought out my plate, and my plate had um, these six um, kidneys around the perimeter of the plate. They were like quite big, and I looked at it and I thought, I'm not going to be able to eat that. And Spencer was such a great person to go out with. And straight away he said, oh, I'll take that, Cal. And he took the plate from me and we swapped. And I had what he had ordered, which was obviously a lot better than what I had mistakenly ordered. And he ate my kidneys.
Oh, he ate it.
And he ate it. Yeah, he ate the kidneys, and I got to eat his meal. So I was really, really grateful for that. I think he did it. Well, he was just genuinely always a nice guy like that, but I think he did it too because it was my birthday. And so he probably felt bad. And in those days I
That's
was
what
a
you ordered. He must
yeah,
have known
mortally,
what let you do it.
yeah. Maybe. Yes, yeah, yeah, that would be the kind. Yeah, he would. He was sitting opposite me. Either he was deep in conversation with someone and didn't realize what I had ordered, or yeah, he thought it was probably amusing at the time. But he ended up eating it for me, and I had whatever he had ordered, which I was really relieved about because there's no way I could have eaten that.
Actually, I don't think
But
this is.
in those days,
No, that's
when
not
you're a
the
junior
same picture,
postdoc,
is it?
no, it's not
That
the
is
same
not the
picture.
same picture, so don't worry. Oh,
In those
so
days when you're a junior postdoc, you don't have much money. And I remember I was living pay to pay. And so there's no way I could have like ordered another meal. Like that would have been
yeah.
it.
Oh. Okay, I'm going to ask just while we're on it. What is your favourite memory of Spencer then?
Mm, uh, I've I've got a number of really good memories. Oh, like I've got a lot of good memories of, early on watching him build what he did. And, one of the great things about Spencer is that he would always have these really amazing microscopists visit. As I mentioned, he would host these conferences, and then, or he would host these, quite well-known, famous, even Nobel laureates who were famous in microscopy, and they would come to visit, and then we would go out for dinner, and he would always invite us along to those dinners and make sure that we got that opportunity to meet those people. He was always very inclusive, and I think I do have one really. The most recent memory is one of my favorite memories, I think, and probably because it was very recent, which I can tell that story. So we I used to come back to France every year because my husband's French, and we would always catch up. And it was a couple of years back, I think it was in 2023, I came back to France and we invited Spencer to come to a party with us, and they were doing this orienteering, what do you call it when you get clues and then you have to go find things?
Orienteering. Yeah.
Yeah, and it was sort of designed to enable people to get to know the region where we were. And then there was myself and Spencer and another friend of mine from Melbourne, and our daughters were there as well. My friend had her two daughters, and I had my daughter there, and off we went in the car. And we had these clues that were written in French, and none of us could understand them, not even Spencer, even though his French was, really, really good.
Yep.
But he couldn't understand the clues either. Anyway, we eventually ended up in this small castle, which was not too far from the house. And when we arrived, all the gates were open, and there were no cars parked inside, but we were wondering, is it open to the public? Can you go in and have a look inside? And we drove up to the front door, and there was nobody, and the door was wide open. So we just thought, and I was really skeptical that it was possible to go inside. I thought, I'm not sure if this is like open to the public. But Spencer and my friend and the girls, they went inside, and I stayed in the car because I was too chicken to go in. And I stayed in the car with the engine running, ready to
Like
make
the
the getaway.
getaway driver.
Yeah, the getaway driver.
Yeah.
Yeah. And Spencer, and off they went. They went into this small castle, and I was a little bit nervous. And they came out maybe 10, 15 minutes later and jumped in the car, and off we went. And it turns out that that's where they filmed the Hunger Games. It was like is it Governor Snow or something, like it was they filmed in, so they did do some filming for the Hunger Games there, I heard, but it was definitely not public. It was a private residence.
And they just
So
went.
so we used to laugh about that. I just found that so funny that, Spencer probably kind of knew that it was possible that it was not public, not open to the public, but I just loved that kind of I guess, that he was sort of willing to, they didn't go very far. They just kind of stepped inside the door and looked at it, because it was this neoclassical kind of architecture, and the way it was all designed inside, all the furnishing, and, but it was funny. We used to laugh about that. But yeah, in hindsight, I wish I had gone in with them.
I know. I'd rather see the video footage of we're after the getaway driver that was
Yes.
actually, you just sat there petrified.
And by the way, we never succeeded to uncover any of the clues. We ended up getting back to the party. Everyone had been back there for quite some time and were having their aperitif, and we kind of missed out on the aperitif, but it was fun. It was fun.
I'm
That's
going
a
to
good memory.
change subjects in a big way. I asked you what you wanted to be when you're young. We worked out how you got to where you are. If you could do any job in the world for a day or a
Yeah.
week, just to sample what that type of job is, what would you do?
I would love to be, now I think more and more I would love to be a doctor. I'd love to, I wish I had have studied medicine, or, I think it would be fantastic to do something like that. Yeah, I often think about that. I think at the time I didn't feel that I had the confidence or the maturity to do something like that when I was younger. But yeah, maybe that was a bit unexpected, but yeah, I would, I sort of, I think it would have been a great pathway to take, which is
Okay, so some
similar to
quick
some extent.
some quick fire questions for you. Are you an early bird or a night owl?
Early bird,
PC or Mac?
PC,
McDonald's or Burger King?
McDonald's.
Hong Kong or France?
Uh, I'd say France now.
Ooh.
France for sure. Mm.
To Australia?
Sorry, say that again.
France or Australia?
Oh, yeah, that's a tough one. Um, that's a tough one now. I sort of feel a bit divided on that one. Yeah, probably
Okay.
Australia, but that's yeah, taking the easy path probably.
Gold Coast or Melbourne?
Melbourne,
See, you have to see. You
Melbourne.
should have said, yeah, you should have said Australia. You should have said Melbourne because that's where you're employed at the moment. Yeah, they'll have you.
Yes,
So,
yes.
uh, beer or wine,
Wine.
red or white?
White now. That's a new thing, but I prefer white. I find red too heavy for me now. Yeah.
Okay. Chocolate or cheese?
Oh, chocolate.
And so, what is your favorite food? If you were to be taken out for dinner
Uh,
what is your best thing that they could put in front of you?
oh goodness. What is it? Confit de canard.
Okay.
Oh, it's probably going to be controversial. Although I might regret. I really, at the moment, I've got this thing for Thai beef salads.
Okay, that's a good shout. What about
Hmm.
the opposite? What is the worst thing someone could put in front of you to eat? Oh.
Brussels sprouts.
Oh, not kidneys. I was going to say half-cooked kidneys. I thought I've just
Oh,
asked a
oh gosh.
question.
Anything that's kidney, brain, or anything like that would be the worst. Oh, I know, I know. I can never pronounce it correctly. Andouillette. Andouillette. It's made of like intestines and it gets wrapped up like it gets rolled. The intestines get rolled and then they slice it. And it's just like a roll of intestines. Yeah, that would be the thing that and bouillon. I don't, yeah, I don't want that served up to me.
Quite literally, that sounds awful.
Yes.
Bet that's a bad day. Oh, you'd be gutted if that was served in front of you, wouldn't you?
Pardon the pun.
So
Yes.
put a few sprouts on the side.
Yes.
Right, when are we meeting up next? I know what to serve. A
Yes.
Thai salad.
Or
Okay.
chicken feet is another favorite. Chicken feet is the other favorite that I have, yes.
Okay.
Which I had in
So,
Hong Kong.
do you prefer to eat in or eat out?
I prefer to eat in now.
And do you
Yeah.
prefer to cook or wash up?
Cook.
So, what's your signature dish?
Oh, what is it? I've been doing this slow cooked lamb. I think that's a bit of my, a bit of a signature dish at the moment. So I really like that.
Uh, Anything that's sheep related and with yourself, I'd just be really wary. People want to try my slow
Yes.
cooked lamb. Oh, yes, please. Aha, I kid you not. That's what it is.
Yes. Yes. The
Uh,
kidneys. I'll never forget that.
book or TV?
Oh, probably TV. I don't really watch much TV, to be honest with you, but I don't read many books either. I think because if I do any reading, it's usually paper, scientific
For work.
manuscripts or papers,
Yeah.
and that's where I spend my time. So that would be my preference, reading scientific papers over. TV and books. Books are better, but TV is that thing that you go to not very often, but when you just really just want to
Switch
lounge
off.
and relax and switch off. Yeah.
Do you have any TV vice, any trashy TV that you like to just to to shut off to and just entertain yourself?
Uh, Not really. Nothing I can think of. Started watching an Australian TV series, which is quite good. I think it's called Upright. So if you want to look that one up, but with Tim Minchin, but other than that, I can't really think of any sort of. Oh, sometimes I probably do go down the trashy TV path, and I've been known to watch. I'm just trying to think now, those reality TV shows, but it's not very often. It's very rare. Just
Okay.
for sometimes it's good for a good laugh. Yeah.
What about your favorite film?
Oh, favorite film. I probably should have been prepared for this, shouldn't I? Oh my goodness. What's my favorite film? Oh, I don't know. Of all time?
Yeah.
I don't know. I'm going to tell you something, but then I'm probably because it popped into my head, but I don't know why it popped into my
Yeah,
head,
go
and
for
it's
it.
probably not really my favorite film, but it's Dirty Dancing.
That's more trashy than your TV.
It is, it's totally trashy. I don't even know why I thought of that. I'm just hopeless when it comes to remembering any films, this sort of thing.
No, that's a good answer. That'll be a childhood
It's
film,
a
though.
really old one, by the way. I
Yeah,
think it's like from the 80s or something.
that'll definitely be one from the childhood times. What about Christmas? Do you have a favorite Christmas film?
Yeah, that would have to be Home Alone is one of my favorites. We always watch it every year with the kids, and
That's a good song.
we love it. Yeah.
And favorite music or style of music?
Look, I love Jeff Buckley. I love lots of different types of music, to be honest with you. I love listening to the 80s. I usually listen to
Mhm.
80s music. I love Leonard Cohen. I love ugh, yeah, I'm bad, aren't I? I can't even think of
No, it's fine, Kelly.
I don't usually go for one artist, if I'm honest with you. Like I
Yeah,
have a select
one was good.
yeah, album of the different songs, and I sort of like to play through all of them. And most of them admittedly come from either the 80s or the 90s.
Okay, so an easier one. Favorite color?
Blue,
Not dappy, not herxed, blue. And you call yourself a
blue.
micro. You should be ashamed of yourself for not having a better
I
answer.
know. I know I should have said, yeah, I don't know. But yeah, it's blue, even though it's not even the easiest one to see, right? Like,
Yeah, but I think it's relaxing as a color because I
yeah,
think
it
that
is. It
you don't get
is.
the same intensity.
Yeah.
Um,
I think I
on
always
to
go
yours.
back to this one. Yeah.
What
Sorry.
about your favorite conference?
My favorite conference right now, or has been for some time, is Seeing is Believing at the EMBL, which happens every 2 years. And I really love this conference, and I'm going to it this year actually. That's one of my favorites. I just because you get to see like really cutting edge applications and of, pretty advanced microscopy techniques. So I really love going to this one. And I often run into people that I know that I've worked with in the past. And so that's a lot of fun.
What about you just come back or not long been to CTLS, Core Technologies for Life Sciences? How was that?
Oh, it was a great meeting. It was a really great meeting. You know, what I love about this meeting is that they cover all things that are really relevant to those of us who are working inside of core facilities, so, career pathways, how to manage core facilities, how to set budgets, how to balance, like you were talking about earlier, how to balance like that, your research that you might, any kind of R&D that you might be doing with you know, more basic services, routine services. And it's always great to get different perspectives. And so I did enjoy this meeting. I don't usually go to it, but I should go to it more often because I think it's actually really great to speak to people there.
Yeah, and again, Spence was very instrumental. That was his baby that he set up and got running.
Yes, it was.
Again,
Yeah.
another legacy that Spencer has given us. Uh, just thinking on that, the different balancing and the work and all the different aspects. What's been, do you think, most challenging aspect to your job as it is today?
The most challenging aspect is, look, as you say, being able to balance different priorities. It's I've got some really talented scientists in my team, and so the R&D component is really important for them, I would say. So I get them engaged in research as much as possible because it's an opportunity for them to continue to publish papers and also feel like they're really contributing to the research that's going on at the institute. I think if I wasn't able to do that, then it would be really hard to retain those scientists. And so, what's challenging is really how do we continue to fund that and how do we make it sustainable? And yeah, that's the thing that I guess I find most challenging, but that's also the thing that's most rewarding and most I want to make sure that I can provide those pathways for the people in my team, and then one way to do that is to give them those opportunities, and then to support them. If opportunities for them to manage their own facilities come along, then I would absolutely support them to do that. But I want to give them that good foundation, and that means that they kind of need to have that broad experience where they're not just necessarily working in a core facility, maintaining equipment, training individuals and working with them even collaboratively, but they have an opportunity to also really contribute to some of the R&D that we do and contribute to papers. And so and that was one of the things that Spencer taught me, actually. And that was what I observed in Spencer's lab. Really, early on, he created a team that had those opportunities, and he was always very inclusive and generous in that way. And, I think that's, the fact that he had team members that he had working with him for a very long time. It's kind of a testament to that. But they also, a number of his team members went on to manage their own facilities as well. So including myself and Olivier Renault and Joe Dragavon, who you might know, who's at Boulder in Colorado, and Natalie Olner and even John-Yves Tinevers, who now manages a sort of image analysis team at the Pasteur. So I think Spencer's had a lot of people who've come out of his platform who have gone to other places to carry on that tradition and that legacy that he created. And yeah, so I think that's really important.
So the next question I have is, what would you say has been the most difficult time in your career?
The most difficult time in my career, there's one that I won't speak about, and that was during my postdoc. And then there's the other one I would say was when I was making that transition from head of the facility to kind of laboratory head, and being able to have now an opportunity to do research as well. And at that time, we also transitioned from being a full cost recovery facility to being one that was subsidized. And there was a lot of, when we were a full cost recovery facility, it was just myself and Lachlan Whitehead, who's a physicist by background. And Lockie was kind of handling all of the analysis side of things, and I was handling most of the microscopy. And we were really run off our feet. And we were, probably at that time we had about 200 users. We were running around. We were working with equipment that was probably suboptimal at that time. We didn't have a lot of funding. There wasn't a lot of investment being made into microscopy. And so there was always a lot of investment being made in flow cytometry, but not so much in microscopy. So that was a really difficult time because the expectations of the researchers were not matched with the investment that was being made. And if I look back now at what we were able to accomplish with no investment and no subsidy, it was very little, I was spending a large amount of my time writing grants to get $20,000 so I could just get a new computer and some software. And I don't think people within the institute could appreciate how hard that actually was and how little resources we had. And it was sort of in 2017 when I was made laboratory head, they also invested a lot in imaging, and that's when I was able to recruit more staff, and I was able to invest in many more microscopes that allowed us to diversify what we were able to do, and that completely changed what we were able to achieve. And I think that's the difference between no investment and investment, and the difference is it's extraordinary, the difference that it made. We were publishing, co-authoring, we were publishing papers a lot more. We were, contributing a lot more. People were a lot more content. With the researchers coming in and using our facility, we're a lot happier. And so it really did transform what we're able to do.
But can I push back on this? Would you say that difficult time was beneficial to you long term because you got a better appreciation of the value of what's coming in and the staff resource, and not to take it for granted? If you'd have had that from the start and it was just gifted all the time, would you be as lean and as productive as you are now, having known what it's like to work in really difficult positions?
Oh, yeah, no, you're absolutely right about that. Yeah, you're completely right about that. I wouldn't have understood that if I hadn't had that experience. And, I would say that experience probably was about half of the time that I've now had the opposite experience. And so I do appreciate that enormously. And it's interesting because when I went to CTLS, people were speaking about that. And most facilities don't get subsidized, right? And most facilities don't get that injection of funding. And some do, and not all. And so I can see and appreciate how difficult it is for those facilities. And the only advice I would have is that, I worked really hard to find all those little grants that you can get. Sometimes they were $10,000, sometimes they were $20,000. The Victorian Golf Club, for example, gave me $3,000, and I used it to, I think I used it to buy a computer or something like that. But, it's just taking advantage of those opportunities as much as you can. And I think little by little, we were able to demonstrate why we needed more investment. But yeah, definitely we run a pretty tight ship, I would say.
What changed then? How did you get this windfall? How did this position change that now you are subsidized and you do have equipment coming in? What was that trigger? Was there a trigger?
Yeah, there was. I mean, there was a couple of things. So there was one research team who were getting heavily into microscopy. They had recruited a very good microscopist who came in and did these fantastic 3D images of mammary gland tissue, and spectacular. And I think they became really heavy users of the sort of only one confocal that we had at the time. And I think they were doing these big images that they would tile scan, they would put on at 5 o'clock in the evening, and then they would run it until 10 o'clock the next morning, and just acquiring a big tiled confocal image, 3D image. And I think they were starting to, they had a paper that came out in Nature, and they were starting to produce these really spectacular images. And at the same time, and so they were demanding that we had more resources. So there was a push from the research teams inside the institute. But at the same time, WEHI had this cancer drug that was highly successful. And so they had a bit of they had some funding that came in through that. And so the Institute was looking to reinvest. And that's really like everything just fell into line, and the stars aligned, or whatever you say. And I think we got this big injection of funding because what they could see was that the imaging facility really needed to be investment there. And so from that, I was able to recruit an engineer or biophysicist from the University of Glasgow, Niall Gagan, who came in and he built our lattice light sheet microscope, which we have, and it really changed a lot what we were capable of doing. Yes, this is it.
So those who are listening can't see, but I've just put a picture up of the partly built lattice light sheet.
Yeah, partly built. That's right. Yeah, that's Niall on the left, on my left, in the brown jumper, and that's Lockie in the blue shirt. And Lockie, he's a physicist by background as well, and they Niall was responsible for building it, but Lockie really worked closely with him, and in fact, Lockie got the opportunity to go to Genelia and work closely with Eric Betsick's team to learn how to build it. And so that was, and him and I both had been there a couple of times. Yes, that's my donut. So I got to also go to Genelia and use the lattice there and work with Leong's team.
But Kelly, you went there to do the lattice light sheet, and yet a donut comes from a STED microscope.
Yes, but it's only half a donut. Yes, that's right. Yeah, you're absolutely right. That was one of my favorite things about Genelia, was like they would have donuts on a Friday or something like that. And that was great. Best donuts I've ever tasted.
There was another picture. We've only got a few minutes left. And is this? Please go on.
So this was in 2011. We opened up a new facility, and this was the first confocal that we invested in, and that's the Zeiss LSM 780. And I probably reached out to you at that time to ask you for advice about it. But this and these two individuals here were partly donated to the purchase of this microscope. So they were from a foundation in Melbourne, and they contributed some money towards the purchase of this microscope. That's why we're having the photo together. But the LSM 780, we installed it in 2011. We retired it in about 10 years later in 2021, I think it was, or 2022. And Zeiss told me it had been used more than any other confocal of theirs in the world. I think it had done like 30,000 hours or something like that.
That's a lot.
I think they had done so many. Yeah, I think it had done so many hours that they had replaced like almost every part inside of the scan head by the time it was kind of getting close to its end of life. But it was a very, very good investment for the Institute.
So I'm going to ask some other just quick questions. So I wanted to ask, who's been your inspirations?
Spencer Short, absolutely, has been one of my biggest inspirations. Lynn Griffiths, who I did my PhD with, was really inspiring as well. And the other person would be Alan Kalman, who I've worked very closely with at WEHI. He's well known in the malaria field, in malaria research.
And what hobbies do you do to relax out of work?
My favorite hobby is basketball. My daughter plays basketball. I love watching her play. I love going along to the games. I've just signed up to a subscription for the WNBA, believe it or not. I never thought I would ever do that, but I love watching the WNBA. the women's National Basketball League in the US. And so sometimes I have late nights watching those games. Other hobbies would be, getting out, riding bikes, traveling, going to the beach, and yeah.
Oh, to be in Australia. Kelly, I have to
Yeah.
say, we are up to the hour now. So, Kelly, thank you very much for joining me today. Thank you, everyone who's
It's
listened.
been an absolute pleasure. Been a pleasure.
Thank you, everyone who's listened or watched. I can't believe how many previous guests you've name dropped, from John Eves to Tinevez to Jason Swedo to Kurt Anderson to Jennifer Lippincott-Schwartz, all dropped in your conversation within it. So if you've enjoyed watching it, please do subscribe, or please do share it with friends as well. And Kelly, thank you very much. You've been brilliant to talk to and to catch up with, and I look forward to seeing you soon.
Thanks a lot, Peter. It's been a pleasure. Thank you.
Thank you for listening to The Microscopists, a bite-sized bio podcast sponsored by Zeiss Microscopy. To view all audio and video recordings from this series, please visit bitesizebio.com/the-microscopists.